Dr. Andrew Harford is the team leader at Water and the Sediment Quality at the Environmental Research Institute of Supervising Scientists, ERISS. It's a mouthful. The Supervising Scientists protect the environment and people from the uranium mine impacts of the Alligator Rivers regions, and as the rehabilitation of the Ranger Uranium Mines advances, the Supervising Scientists are developing a long-term monitoring program to ensure stakeholders' expectations are met. And today, Andrew's going to talk about the genomics aspect of that. Please take it away, mate.
Thank you. And it's great to be here. I've been a long time adjunct of the school and CDU, and you know it's really important for us to get up here. This was a presentation I did for our technical committee. They asked for it, and I presented this in May. And I thought, well, what a great opportunity to get out and talk about it with a wider community. Maxine, I saw do an eDNA talk, and so there's a bit of assumed knowledge with this. I'm not going to go into the nitty gritty of what eDNA and genomic technologies are, so hopefully that's all right. And it's a bit of a high level management overview, really, all the different things we're doing and our paths for the future. So not a lot of science, but what I would propose is as we get some of the results in for some of these big projects, that I send up some of the team and they talk about these awesome projects that we've got going on. So let's get going. So I'd also like to acknowledge the Larrakia people of the lands where we are at the moment, and the Mira people of the Jabiru area. They're the traditional owners of the Ranger and Jabiluka mines, and also the Binning Mungai people of Kakadu. We work on their land, and we pay our respects to their elders past and present. So it did occur to me, I get fun questions all the time about the Office of the Supervising Scientist, especially when you're at conferences with this little Supervising Scientist logo on. If you don't know about it, I mean, I'd hope most of you would, it's a fascinating part of the NT history. So we were established in 1978, it's a statutory office, and it happened after a review, we call it the Fox Inquiry, and it was a big review about the environmental, social impacts of uranium mining in the Alligator Rivers region. A lot of things were happening at that time. We were establishing an NT government. Uranium was going to help bankroll that, essentially. They set up land rights, they set up Kakadu, all these things happened in the mid to late 70s. So the big mine that's been, you know, the producer is once the third largest producer of uranium oxide in the world is the Ranger Uranium Mine. And that's surrounded by the World Heritage listed Kakadu National Park. And we've been there deriving water and sediment guideline values that we use them for operations, but also now they're the targets for closure. Whoops. We also conduct a very substantial annual monitoring, both of the surface water chemistry while the creeks are flowing in the wet season, but also the biology during the recessional flow period. So the mine site, you probably know, is in rehabilitation. It's a big job. They're talking over $2 billion worth of rehabilitation. So the company is required to return the land to the Mira people in a state that is suitable for traditional practices. And because of that, there will be a long-term monitoring program to look at the protection of human health and the ecosystems. So our focus of research, a big focus of our current research at OSS, is the development of monitoring methods using these really awesome key enabling technologies, things like drones, satellites as well, AI and machine learning for image analysis, and of course environmental omics, also called eDNA. eDNA is not a term we started using. We were using omics, but since then we've been explaining a lot how it relates to eDNA. Not everything is eDNA, so we use that omics term. So why did we invest in omic technologies? Because it has been a decent investment. So we do have this responsibility for the long-term monitoring of the Ranger mine. And we've had this great history of evolving what are leading practices into best practices. And that's had national benefits. Things like the Australian New Zealand water quality guidelines, the Ranger Uranium Mine has been a test bed for those best practices that are used across the country. We've got this annual integrated creek water monitoring program. We look at macroinvertebrates, fishes, aquatic vegetation every year. And at the same time, our ecotoxicology work, developing those standards I was talking about, we've got all the standards that we need. We're making decisions about closure right now. So that was winding down and we had some capacity to spare. And senior management was asking me, what are we going to do with all these great people? On top of that, genomic technologies had got to a point where they had this promise, but that promise was really becoming a reality of fast, cost-effective, safe. You know, being able to dodge crocodiles is a key risk that we have. Oops. Might have a time thing going on. And so that was becoming a reality. We'd been using these tools for about a decade before we came to this conclusion of let's invest. Other institutes were developing strategies. If you have a look at NOAA's Enabling Technology Strategy in 2019, it was about the same time we came up with our strategy. Commercial entities were already selling eDNA as a way of doing environmental monitoring. Our friends at EnviroDNA, they started with platypuses out of Melbourne University. They were selling their wares as early as 2015. And we also saw the formation of these professional societies, such as the Southern eDNA Society. So you know, they were established in 2024, but around 2021, 2022, we were talking with a group of people. We called it the management team, and that eventually evolved into a proper board and professional society. So this was our journey. So I love that photo because those sediments are a pain to try and measure any sort of biology. Trying to count bugs in it were a real problem. So it was about that time we met a guy named Anthony Sheridan, who's been there throughout this journey. And he came in and said, we can use DNA to measure what's in your sediments and establish the impact of spiking uranium into those sediments. We're using a technique called 454 pyrosequencing. So that shows how early days it was. And we had a great PhD student, Brodie Sutcliffe, who did some microbial analyses, really looking at the function of things. And we had our first publication in 2017, and she went on to publish a lot more. And have a look at her papers, they're fantastic. Lisa's PhD with Jenny as a supervisor, Grant Hose from Macquarie University, our friend, also helping out. So she did a great job at looking at groundwater ecosystems, and it was really important to be able to do that by DNA-based methods because counting stygofauna was really hard as well. Whoops. So we went to genomic techniques. So we started barcoding things in 2017. We knew we were going down this path. We had an inkling. So we started barcoding things with a grant going to Macquarie University. And I'll talk a bit about our barcoding strategy as we go along. We developed our genomic strategy in 2020. That was basically our internal pitch to senior management saying, this is what we want to do for the future. And it worked. 5 years on, we've got a really healthy program going. We really only started collecting macroinvertebrate samples for some of our big projects in 2021. So what are the opportunities? I mean, this is just about the most basic stuff about eDNA. So DNA traces are everywhere in the environment. We can find them. Oh, how do I start that? That's a video. There's DNA in the groundwater, in sediments, in water, soils. Air is a big thing at the moment. And we've been looking at in parasitic organisms such as mozzies. You can get it from leeches, flies as well that are feeding on detritus, but also whole organisms is another place you can get your DNA and do identifications rather than under a microscope, but with a sequencer. And it's also promising this whole of ecosystem assessment. Take a sample of water, do everything. We're not there, to be honest, but that's the promise of it. So fast, effective, whole of ecosystem type things. So there's a lot to do. There's too much to do. So this is our focus. This is where we have, taken our bites of that, those different opportunities. So this is our aquatic monitoring program. Erica would be very familiar with this. This is what I deliver to her course. I talk about this monitoring program. So we look at the chemistry, we look at the biology, we have our wet season measurements and our end of recessional flow assessments as well, where we go and look at our macroinvertebrates and fishes. This is on the terrestrial ecosystem side of things. So this is a representation of the state of transition model that really guides the revegetation of the mine site with help from Anna Richards. She started that project a little while ago. So we've decided that we will look at our macroinvertebrates because that's our key monitoring tool for determining if there's an impact at the end of the wet season. We're getting into the fishes with help of Macquarie University PhD. But on the terrestrial ecosystem side, we're looking at nutrient cycling, and we've also done a bit of work in the vertebrate, and Ali's doing a project on invertebrates, but we'll get into the details of that. So that's just a bit of an overview of our two big monitoring programs and where we've decided to focus for now. So it's not been without its challenges. We started in about 2020. We did that review, that strategy review and a plan. You might remember 2020 was the COVID days. So there's, despite all COVID and things like that, there were some other things that we learned along the way that I'd like to share. So we had a bunch of ecotoxicologists and ecologists. So we had to reskill them to become molecular biologists and DNA chemists. Some of them had no experience when it comes to DNA chemistry. The time to take to really get them up to speed, we underestimated what that would take. We had access to expertise issues, people not being available, coming and going. So that meant more mistakes, less guidance, meant we had to rectify things along the way as those people were learning. We have this big monitoring program. It's our number one job is to provide stakeholder assurance that there's been no impact from the mine. So every time the recessional flow period comes along, Lisa gets really busy, especially being the manager and organising all that. So we basically down tools on the R&D for a significant part of the year. And we've also had other priority projects come along and distract resources into that. We've had some toxicity testing. We thought we were done. We went back and did a little bit of aluminium toxicity testing, which meant some of the technical staff that were on genomic projects were now back into the tox labs. We had to build a lab, so that was not without its challenges. So in purchasing all the equipment, all the things that we needed came in slowly over those 21, 22 years. So on the extraction side of things, we started with the most complicated stuff. I don't know why, we just sort of went, let's try and do this sort of, and I'll talk about that, but it was really difficult, especially when you had inexperienced staff. Because they were difficult samples, we also had to troubleshoot and go to non-standard extraction methods. We weren't using kits anymore. We were actually going back to basics and making kits from scratch, which was great and needed. So yeah, we had to go down some interesting paths. So the original methods using those kits, we had a lot of failures. Half the samples, we need to re-extract those to make things work. There was a lot of troubleshooting with PCR, there always is. But when you've got staff that are just learning, it's going to take longer. Sequencing during COVID was awful. We had 12-week turnaround times being told at some stages. So we were waiting for data to come in, and that was really delaying progress. Bioinformatics has been a big challenge. That when I talk about access to expertise, that's been one key area. We're building up internal capabilities. We've got coders that don't understand DNA chemistry that we're teaching how to do their bioinformatics for, and that's been needed, and we're very happy with building that expertise, but that costs time. And of course, while we're waiting for final results, it's hard to progress on and make decisions on the future for other things. So that was some of the challenges we faced between 2020 and now. I want to also say we're not doing this in a bubble. We've got a great network of people. Anthony Sheridan, like I mentioned, he's been there from the start. He's been our general advisor while building this lab, but he's also gave us some really fantastic PhD projects that I'll talk about. We've been collaborating with David Beale on metabolomics. He's amazing at that. He's online. G'day, David. And also Andrew Bissett, really great. He runs the Australian Microbiome Initiative. He's been helping with some soil microbial analysis and putting anyway, I'll talk about that soon. Ali and Sam were helping with the DNA barcodes for for fishes and others. We did some other things, but mostly vertebrates. And that was a long time ago now, really. So that was Ali's master's project, and now she's doing a PhD with the help of Sam, Maxine, and Alan on terrestrial invertebrates, ants, and termites. I'll speak about that. I've got this great. I haven't really been that helpful. I've ran out a bit of time, but Jeremy, Andrew, and Jay have this great project. I'm a collaborator on that. I'm really hopeful that when the time comes, I'll be able to use that, well, you know, collaborate with that project and get some of these technologies and the data sets into their platforms that they're building. Michael and Peter Dostine have been a great help when it comes to identifying fishes and macroinvertebrates, and they've been sending their samples and their specimens to us. And we've also got some good links with CRC Time. We've got one particular project that I hope that will help our soils project, but I'm also talking to them about using their relationships with partners and regulators to actually get some of these R&D things implemented to the point that regulators know what to do with them. And I'm really hopeful that will happen, but it needs work from me, and that maybe next year now. So we've got some great professional networks and community practices. The local one, that's a picture of a workshop that we had, a group of really engaged people from the Darwin area. I've got a DQ one that I've not been giving a lot of love, but I'm meaning to pick that up. They're really great to sort of spread the word amongst government departments. It actually started when we were DOOR, so Ag were part of that, and I'd like to re-engage with Ag as well for that. Like I mentioned, the Southern eDNA Society has been a great network of people, some really good people in that group. We've got our potential data receivers. I've been talking a lot to the National Biodiversity DNA Library and the ALA about what we can do with our data sets, especially our barcodes for the NBDL. We've just signed, in fact, if Di's online, she would find out, we've actually signed an agreement with the National eDNA Reference Centre. I mean to tell her, it's no, yeah, we've actually got the DepSec Secretary to sign up to that. So that's great. And I'll talk about why that's important. So thanks to all the collaborators. We shouldn't do this in a bubble. There's so much great stuff to do. And that's why we join into those networks. So a while ago, my senior management came to me and said, I want to know what this strategy is. You've got it in your head. I want it on one page. So this is what I came up with. So I think it's a great tool. So we've got the mission there to develop cost-effective, reliable DNA-based monitoring tools that can be used by the traditional owners and Indigenous ranges of Kakadu to care for country. That's the ultimate aim. So within that, we've got all these different steps of and pieces of the puzzle. R&D, it involves optimization of DNA extractions, PCRs, and things like that. Then once you get that, you've got to put it into the field, design your field sampling, statistical design, and things like that. So once you get that, you take it, you get your temporal and spatial resolutions, you start assessing that for your ecological questions in mind, and really start, you know, understanding what this data is telling you. So a lot of the work is in the R&D space. I'm really keen, and we have the privilege at OSS of being able to do these long-term projects where we can push it into implementation, and then we can have these long-term data sets and really understand the value of these protocols. It's a real privilege to be a government scientist, and that's one of the reasons why. So of course there's a bit of back and forth. So there might be times where you go, all right, I've got a field design, I've tried some things, I want to go back and look at different genes. I want to optimize because I've got more barcodes and things are developing and things like that. Likewise, you get into the field, you're looking at your spatial temporal trends. Well, I need to change up my field design. So there's this back and forth as well. This one, extend capabilities. This is all the fun stuff in the future. Things like, you know, long reads and all that. I'll talk about that at the end of the presentation. But there's some really good things that will mean that this will always sort of be circular. That we will get to a point where it's like, all right, we need to change it up. PPR3 techniques, for instance, would be in this extent capabilities. So essentially for all this, we want to put that First Nations participation. We're pretty good with our consultation and needs and things like that. But at the core of it is actually getting a system that will work for them where they can do it for themselves. So that's central to that. And because I'm a scientist, I couldn't do it on one page. This is the second page of the actual thing. This is all the project. Well, it's all the ecosystem components of where we're at with them. I'm not going to make you read this. This is what I'll step through now. So it's all the different sort of tasks that you need to get through that process, and all the different ecosystem components that we might do. Summed up on one page. So barcoding strategy, this has been a big focus of ours. We know that it's really going to be helpful to us to unlock our identifications and understand ecological processes, but we also know that it will be benefit of others. So we want to do this, and we had the barcode workshop last year because of that. So good barcodes enable accurate species identification, and then that unlocks a deeper understanding of ecological processes. You can also use them to sort of mine them. Ali did a great job of this. You've got these barcodes. Alan has a wonderful barcode library for ants. So she used that to design the primers and make it better rather than just pulling something off the shelf and going, that's it. We've been focusing on mitogenomics to future-proof things. It costs a little bit more. When we started that project in 2017, we almost didn't go down this path, but I'm so glad we did, because you get 37 different genes from a mitochondrial genome. You can skim, I won't get into the technical details, but it's a great way to get a bunch of genes. You don't get things like the 18S gene, because that's a nuclear gene. So we did this. Ali's, like I mentioned, master's thesis for the aquatic vertebrates with the help of Michael and the museum giving us samples. And now we're actually using that library through a PhD at Macquarie University. So aquatic invertebrates are still a big focus of what we're doing. We've got a few hundred species that have been shotgun sequenced. Some of those ones that didn't work for that genome skimming, we went and we Sanger sequenced. So that gave us 3 genes. So, you know, we might go back and revisit some of those. And there were about 80 that didn't work, I believe. So we need to go back. It's very hard to get good quality DNA out of some of these really small things. We under-resourced the vouchering process. So we don't have voucher species so much, we have voucher sheets. And we've been talking to people like the NBDL about the appropriateness of that. We think that it's a good way of doing it, because honestly, when things get so small, we're going to destroy them, right? So we've gone down this process that's a little different to what they would like, but it's also taken a lot of time. Tegan, we've got from CDU after finishing a PhD, and she's taking photos for us still. So we've done all the important sensitive groups. We're moving on to some others. Taxonomic experts are always going to be an issue. They're a very rare species. And that's what we found when we had a chat about that at this workshop. We did it June 2024. We really wanted to consolidate everyone's ideas about what they needed and what we needed in resourcing, i.e. taxonomy, to make sure that it works. We got a nice document out of that lists all the outcomes. That's on the Sedna's website. Thanks to them for their support. And thanks to CDU and NT government for their support as well, to have that discussion. So there's others collecting barcodes, CDU, Magnet and things like that. So it's really important to sort of consolidate, strategize that, make the most out of it, and just have everyone understand where things are sitting. Well, the workshop showed that there's some groups who are going to have some real challenges. Stygofauna, I know Jenny and Erica are very interested in that idea, but the smaller things, the hard to find things are actually going to take some time. We recognise that in this document, that's going to take some resources. Very important to do so. Like I said, groundwater is the best way to do it, but we need to come up with resources to get the barcodes to actually really unlock some of those ecosystems. Oh, data governance and management. So a constant thing that we need to work on. And, you know, we've got some good systems, but we need to make sure our data sets align with those FAIR principles. Nicely, if you have a look at CSIRO, there's this FAIR E project, so FAIR data sets for eDNA. We had a chat, the Biocommons group, NCRIS group, they got together and we started talking about these data sets the other day. But we have a real focus on making sure that that our data sets are fair. We've got some good processes for the standard things that we do, sample registry, data management plans, but you know, we've got new data sets, so we need to keep an eye on the things that we need, the quality control, quality assurance reporting that we need to do. Bioinformatic pipelines is something that we're discussing right now. We have some real problems with the queue, with just computers. It's, you know, if you're there for any amount of time, you do realise that their focus isn't necessarily on our specialised scientific needs. We're a very rare breed within the department. So asking for things like cloud computing, Azure access has been a bit of an issue. We had some cloud storage computing support when we were under DOR, but that's since dropped off under the queue. It's still part of my future vision. I think it's the way to go. Cloud computing will really help us. We've got some, you know, computing challenges at the moment. Tom ran a data set the other day, it took 3 days, and it's very hard to wait 3 days to rerun and tweak that little parameter. So there's things that we need to do when it comes to just data analysis as well. Like I mentioned, there's ongoing discussions with the NBDL, ALA, about hosting our data sets. And so we need to make sure that our data sets are fit for purpose so they can take them. I had a good chat to Michael about even the barcodes that we did for the vertebrate barcode library and what we might need to do about, you know, getting it into the NBDL. Bit of work to be done, he was telling me. QA/QC, I mean, this goes to FAIR principles, but I thought I'd mention specifically, we've got best practice documents. We helped co-author those. They came out of that University of Canberra group. Really good guidance for how you should develop things. Devil in the detail, but really good. Ali's about to submit a paper following this protocol, and it was such a good job, Ali. Really happy to see you doing that. We've just signed an agreement with the National eDNA Reference Centre. We think it's important to be part of that network. They're really focused on providing support for quality assurance, reference materials, proficiency testing. We want that. It was set up, the National eDNA Reference Centre, to support biosecurity, and you know, they have some really strict legal requirements. If these things end up in a court of law, you know, how do you defend it in a court of law? So we are still looking at our routine QA systems. We need to establish that along with an auditing system. We've learned this from our ecotox projects and program. We need to go down this path for eDNA. And I wanted to mention First Nations capabilities because you'll see when I bring up these schematics that there's certain things that we are doing. We're engaging with the Gunjak Aboriginal Corporation. We do that for all our projects and protocols. So we have some good processes set up for engagement with them. We have the Juruburu Rangers participate in our programs as much as we can. They come out and they help with biomonitoring when they can. But capability building is really, like I said, our mission. So we want things to be, you know, simple, backed by really high-tech underlyings, you know, so that they can go out, take a field sample of water or soil, maybe a passive sampler for air DNA, and then, you know, bring it back, and there's some automated extraction things that I'll talk about that will really help make these things perhaps accessible to rangers out on country. So a simplified data analysis, interpretation, decision-making platform. People are working on these, so that's the vision for the future. So I'm going to hit you with a bunch of these, a little bit perhaps dry, but for each of those ecosystem components, this is what we came up with. I asked the project leaders to come up with these program visions. And over on the right there, we've got a bit of where we're at. So for the aquatic invertebrates, this has been a big investment for us. This is almost a flagship. We've got another one, but it's a really key one for us. So we want to transition our macroinvertebrate sampling, mostly the macroinvertebrate identifications, to a DNA-based method. We want to make it simple, fast, cheaper, etc. We want to improve taxonomic resolution. At the moment, we do family level assessments. So this will unlock species level assessments too. And we need to reduce our reliance on taxonomists because they are hard to find. So we've done 3 years of a side-by-side study. All the data's coming in now. I'm super excited. I've seen a little bit of it, but we're dealing with those bioinformatic challenges at the moment. Actually, I might. So this shows where we are with R&D. It shows that we've got a bit to go with implementation, but we have got these sort of data sets of temporal year by year, 3-year data set happening as well that we will plug into method assessment. Once we get to 5 years, it will really start getting powerful. But I'll flick over to this one because this is the study in brief. We've got our macroinvertebrates that we collect every year. We've split those samples. We've taken them, sent them off to Julie Hanley, who's our macroinvertebrate morphological ID specialist. She's over JCU these days, but was out at Kakadu for 20 odd years. She's really good at what she does. So we took this matrix sample, we call it, that's everything together, including, and these are the ones when I was talking about, these are the most difficult things to extract. It's got all the veg and everything, and it was a real headache to try and even just resolve how to extract DNA from this bulk. This would be a lot less effort than picking the bugs out one by one. We want to reduce our resources. We want to rely on, can we use the bioinformatics to just sort through all that extra DNA that we've got. So to do that, we've got the morphological identifications, we've got the metabarcoding, so that just looks at all the different species using those short barcodes, but we've got metagenomes. So we've shotgun sequenced these picked bugs, so that will let us understand PCR biases, things that we might miss, and a deeper dive into, you know, what the PCR is doing to our data set. And that's really important to understand the pros and cons of the current amplicon methods. Yeah, I did want to say something else, but I forgot. It's left me. A lot of effort. Oh, that's what I want to say. Bloody expensive. So that's the problem with metagenomes. Like, maybe we'll get there with PCR-free techniques as that gets cheaper, but yeah, it costs a lot. So we want to do it once just to understand, and then, you know, maybe adjust, look for the primers that are doing a better job. You'll see here the amplicons, it's not just one gene, we do multiple genes, because doing different genes will get you different coverage due to different PCR biases. So aquatic invertebrates, we've only really just started on this. So we wanted to develop an end-to-end pipeline for processing water samples. Take a water sample, determine our fish. We're focusing on billabongs because we talked a long time. What does it actually mean if you grab a DNA sample in a river? Where did it come from? So we do the billabongs after they lock off. This is our biomonitoring anyway for the lowland billabongs and the channel billabongs out of Kakadu. So once the dry season comes and those are isolated billabongs, we actually do videography these days and some AI image analysis, and that's a great tool. So we've already got a great tool. Why would we do DNA? Well, it will give us a broader range of taxa. There's some fish that just won't show in a video, your sleepy cod and your benthic sample, you know, the ones that like to hide, the nocturnal ones and things like that. So we will get a different data set. We're looking at how that data set might complement our videography. So John O'Shaw, he was up here for a little while out at the field station. He's back in Sydney now finishing up his master's. He's going to roll that into a PhD and he's doing a great job. He's done the barcodes, we did the barcodes with CDU, Sam and Ali, and then we're using that. So the master's project really just looked at the basics. How much water do we need to take? Would 20 litres get us a lot more taxa than 1 litre? Luckily, we don't need to go to 20 litres, but those data are just getting put together right now. The other great thing he's done is he used MinIONs, so benchtop long reads. He did the PCRs, he's got a ton of reads out of that. It's worked really well. So this is why I've highlighted the extend capabilities in yellow over here. We're exploring those, even though we're sort of a bit jumping ahead. It's a PhD, why not? And it's fantastic data. So next biomonitoring is going to come up, while we're doing the videography, he will do a bit of a side-by-side. We'll come up with a sampling design to look at the spatial, coverage of a billabong, and we'll look at what happens with the videos compared to what happens with the DNA in the water. And to be honest, like, the videos still cost a bit of resources. If we can just grab a water sample, then it might be that that's a very much more cost-effective method of doing our biodiversity surveys of fishes. So terrestrial microbial communities, not just bacteria, but fungi as well, archaea as well. So people are doing this a lot already, but I've got a real passion for this. It started through the sediment work. I want to understand function. Like, you can get bacterial structure of the communities, but what does it mean functionally? So this is where we're using this multi-omics approach with David Beale and Andrew Bissett. We really want to unlock that structure function relationship, and there's a good reason for this. Well, there's other reasons for this. We just had a workshop at the CTAC meeting a couple of weeks ago. We do not use microbial endpoints for environmental quality setting because it's too hard, because the guidance says you need ecologically relevant endpoints. So what does a structural change in bacteria mean? They won't use that until we tell them what the functional ecological relevance of that change is. So let's skip over here, because I do love this project because of these reasons, because it's really important, and there's other people that will use this idea. Understanding the structure function relationships of microbial communities is what we're really trying to do. So we've got a bare waste rock. Is it going to be supporting sustainable of these vegetation communities? We've seen some areas struggling. So we want to understand whether it's some of these things. We've got an all-spot style sampling. The great thing about this is we've got all the chemical and biological, the tree veg data alongside it, because we're doing it for our friends in the ecosystem restoration group. And so we're doing amplicons, metagenomes, and metabolomes. Yeah. So we'll get community structure. Thanks, mate. Out of the amplicons. We'll get an inferred microbial function out of the metagenome, because it's still, there might be unviable DNA in there that we're counting as viable, but the metabolome will actually, I said inferred function, as actual function. It should be, we're measuring the actual metabolites that are being produced by those communities. So we want to match it up. Metabolomes are even more expensive and hard. In fact, I put that in there. So we don't necessarily want to do metabolomes, but we want to understand what that amplicon means for function. So, building some model, having some idea of what the way we need to interpret that amplicon data set is really important to us. So, we're hoping it's all, one-to-one, but there might be some adjustments, some steps we need to make to really interpret when we go out and we collect an amplicon data set for and fungi, so important. So people often skip that, but you see it more and more now. The ITS gene's being used for fungal communities. I know Christo out at the re-veg expert at Kakadu Native Plants is big on the fungi, because he reckons they support the trees when they're in their early stages. Terrestrial invertebrates. This is Ali's PhD, and thank you to Alan and Ali coming to us and saying, let's give it a go. Because, our program is pretty full, and she's doing a great job of bringing this to the fore. She's looking at ants and termites specifically, baiting the termites with baits, looking at soil samples and things like that. Alan has a fantastic CO1 library. 10,000, you'll tell me, mate? Yeah, 10,000 ant species barcoded. So that's been a real, valuable asset for the project, and we've been focusing on that. Ali, like I mentioned, has developed new primers using that amazing barcode library. So it shows you the value of having those there. And look, it's not just ants and termites that we're potentially interested in. We actually had Arctic very interested in things that might be affecting early revegetation, you know, some warning signs, but also maybe some indicators for ecosystem re-establishment. Our card's full at the moment, but that's something that we're very interested in. We're watching Sonyu and what she's doing, because the biosecurity team with their terrestrial invertebrates will really help what we do. This might be somewhere where we go to eDNA as well, because that's often, there's a lot of that being done at the moment. Hopefully I can flick through these a bit quicker. I'm running out of time. Our iDNA project. So what are the mozzies biting, and can we do a vertebrate terrestrial survey from that? So Christine Shivers has finished up her PhD, been awarded it. The papers are coming out. We have one in environmental DNA, Kakadu work coming out very soon. It's with the editor for consideration. Look, unfortunately, we have no plans for fauna monitoring in the near term, but you know, we're really excited about this being worked up. I think many other people are already knocking on our door saying, can we use this technique? So yeah, a lot of the R&D done, but implementation, how we do it in the field way and things like that is important. Groundwater community, like I said, Lisa did a lot of work on the you know, R&D side of things. Barcode libraries, they're in red, they might always be in red. So we definitely demonstrated the need and the power of genomic techniques over traditional survey methods. And you can have a look at all the papers for that if you're more interested, or have a chat to her. She loves to talk about stigos. Phytoplankton, aquatic plants, biofilms, we haven't been able to get to it. We had a little bit of a dabble with that. It was a sprint project. We learned one thing, is like, this is not a sprint, this is a marathon. So barcodes, things like that we need. We were able to extract DNA, but honestly, we had some real headaches. We tried to do this for our environmental radiation team that were interested in biofilms and the radionuclides being taken up. We wanted to identify for them. We weren't particularly successful for that. So I'm going to finish up now talking about the future. It's a busy slide, but it's a busy field. So we've got these amazing sampling devices. That little rocket ship thing, the yellow thing, that's a little, you hang it off the back of the boat and it collects DNA samples on a little filter at the side. I want to make it a fishing lure. You know, because there's one thing our rangers out at Kakadu love, and that's fishing. So you can get them to collect samples in a billabong with a fishing lure type thing. That's the backpack that we use. That's the Smith's Root one. Very good, GPS enabled and things like that. But there's also these improved extraction and sequencing methods that are coming along. The Naturometric groups from the UK, they have these systems for, and really what they're trying to do with these end-to-end pipelines. You grab a DNA sample, it puts it through everything, and it's and you can do it all in the field. So this image on the right here, they call that a bento box. It's got everything you need in the one little kit. And that's a MinION. That's how small they are. That's the long read sequences that you plug into a computer and you get. So very field deployable. And different data sets. Long reads will give us a different view of the world. So like I've been mentioning a couple of times, PCR-free methods, that will can't be unlocked as things get cheaper, as the new MiceTech's a beautiful machine. And what's amazing about it is all the technologies in the cells that you put into it. It's not an instrument, so you don't need a lot of technical expertise to keep on going. 100K, 4 times faster, a lot more reads, amazing. eDNA, like I've been saying, that's a hot new field of research. Love to get into it, but we just don't have time at the moment. Capture probes, I love them. So we are looking at whether we can get quality DNA out of mosquitoes for population genetics. So if we can catch enough of the DNA, maybe we can do that. It's pretty out there. We'll let you know if it works. Apps and tools for data interpretation, a lot of people working on that. And that's going to help integrate international infrastructure, such as the ALA. Obviously, bioinformatics, the advent of machine learning, it's very data hungry, but as we get that going, it's going to have the data sets that we need for understanding these genomic data sets. Standardization for regulatory use and biodiversity markets, that's a big thing that I'm into, because that's what we should be doing as government, you know, pseudo regulators or whatever, but we're in a regulatory framework, so we need to make sure that these things are usable for decision makers. Otherwise, what's the point, really? I've focused on genomics, and we have been calling it omics, but there's so many other things. So there's transcriptomics, proteomics, metabolomics, so you can look at your RNA and things like that. I'm really interested in all that sort of stuff. I'm interested in these models, and how these data sets can go into these models is something that's being supported by the OECD and the US EPA. They're called adverse outcome pathways. They look at molecular initiating events. So what happens at a molecular level? Can you sort of relate that all the way through key events that ends up being an ecosystem impact? It's a big call. They do it for human health all the time. There's some very specific molecular initiating events that actually end up in a disease state. So that's where it comes from. So, exciting field, so many things to do. This is us opening up Pandora's box and DNA's in there. So we've prioritised based on what we need, those key aquatic and terrestrial monitoring programs where we can get the most values. Obviously, these things continue to grow. We're partnering with people, we're watching what others are doing, because we shouldn't be doing it in a bubble. We'll get more value if we watch what others are doing. We will need to do engineering work. Universities often don't have the luxury of developing these barcode libraries. We do. We should be doing that for the good of everyone. But I'm also interested in others that are doing it so that we can put it all together. So 2 key projects, that invertebrate, aquatic invertebrates and our soil projects, they're passing that proof of concept stage very soon. We will start building long-term data sets from that. And then we're going to have to transition from R&D to some sort of routine monitoring program. It needs to be time effective, otherwise we won't have any more time for R&D. So I'm mindful of that, and we'll update this strategy on an annual basis. We'll keep you in the loop where we can, and that's it for me. And one thing, thanks to my fantastic team, Lisa, Tom, Kate, Claudia, Mel, Tegan joining the team, fantastic to have them on. It's really important to have the great people that with great wealth of knowledge. Thanks.
It's a great talk, mate. Any questions? I'll let you field your.
Who do I go for?
You've been warned.
Yes, Daniel, I think I know yours.
Thank you for such an enthusiastic and inspiring talk. It was just wonderful.
Thank you.
All that stuff. And to hear it presented with enthusiasm, which is what it's like to be. So the question I'm going to ask you is, I'm going to go right back to your mission statement, where it's all to do with Kakadu. And we know that there are other rivers, the Daly, the Roper, the Adelaide, which are under imminent impacts from other activities that are not uranium. And it would seem just wonderful if ERISS or OSS could now become involved in all of that water quality monitoring and regulation for the territory, because there's nothing else going on that would be equivalent.
And I hope you heard the flavour of that in there. That is absolutely my mission, is to share, to take it out there once we learn from the Ranger Mine, sharing that with others, learning from others as well, of course, and creating forums where we can actually do things. So the CRC time project's a really good example, right? So, and like I said, it's got work, and hopefully Guy Bobs and Jason Kirby are listening, because I would love, they've got the partners there, they've got the regulators there, perfect group of people to actually take, all the opportunities that we've found for eDNA. Like, what have people done? Look at what's value for mine closure and mine rehab, and then fund it across the board where people are needing it, and validating it, explaining that to regulators all along the way, talking about what they need so that they can make their decisions. And so then you get that, it's kind of like Oz Rivers did, right? That's what we need to do for DNA. And that way we can put it on the Australian New Zealand guidelines. I'm quite involved in that. Our group helps that team all the time. So I would love to see guidance on there on best practices and all that sort of stuff. I think that's how ERISS and OSS can best help everyone else. Alan?
I think that's great, really impressive, Andrew. I'm just wondering, you didn't say a lot about what ERA is doing, or the relationship with ERA. And so I was just wondering if you could say a little bit about the relative roles of ERA monitoring, OSS monitoring, and how that sort of fits together.
Yeah, so they've not really picked up the DNA-based monitoring. They are busy with their monitoring programs they'll need for re-veg. Off-site, and what we do with the fishes and the macroinvertebrates, that's always been OSS's responsibility. Other places that's not the case. Usually it's the mining company that will collect that data for an EPA and send it. We don't do that at Ranger Mine. So there's real benefits for them, I would argue, to pick up DNA-based monitoring. I mean, the soil work will be an on-site thing, monitoring that, so they will benefit from that. But there's interest, but there hasn't been a lot of investment from ERA or the Ranger Rehabilitation Project, as they're now called.
Keller. Thanks, Andrew. It's great to hear more about all of the work that you're doing, and I appreciate that overview. My question relates probably to a whole set of presentations that you didn't give today, but with all of the monitoring that's been done, what sort of impacts, if any, have you detected from Ranger Uranium Mine on the aquatic ecosystems?
None, off-site.
Yeah.
And that's been a real success story. I mean, OSS staying out of headlines has always been a success in many ways. So off-site, nothing. So there'll be, and there still is today, a real discussion around on-site. So there's two key water bodies on the Ranger lease. That's Kinjimba Billabong and Georgetown Billabong. They're features that will actually return traditional practices back to that site. So understanding their health status and their trajectory, I think, is something that we're still, I mean, they're definitely working on that. They've got a vulnerability assessment framework that the mine's working on. And sorry, Alan, and I misspoke. They have done some DNA work on site in those billabongs for those. They've done at least a fish survey. And the consultancy doing that, they're very keen to get in those billabongs and do some of these assessments using DNA-based monitoring. So, because understanding what those billabongs, because, you know, Kinjimba especially, it was a mine water discharge point. It's got acidic sediments. So what to do with that is still a matter of discussion. Well,
I've got a question from Sherry Bruce online.
okay.
It says, I'm an Aboriginal mycologist working in Kakadu on Aboriginal ethnomycology and fungi ecology. I noticed fungi only appear implicitly in the terrestrial microbial things you work. Given their ecological importance and cultural significance, are there plans to treat fungi as a distinct focus in the monitoring strategy rather than just background in the genomic data sets? And if so, how will FPIC and Indigenous data sovereignty guide decisions on whether fungal genomic data is shared or restricted? And there's a PS and CARE principles on this.
Sorry?
PS and CARE, C-A-R-E principles on this.
Okay. Great question. So there's a lot in that. So we are looking at the fungal communities as part of being the, you know, important carbon nutrient cyclers. So it's not background so much. We are really understand that they're important. And like I mentioned, Christo from Kakadu Native Plants is very keen on understanding and seeing our data sets and things like that. We don't have a lot of plans, but I did have a bit of a chat with Christo about potential for looking at especially fungi plant interactions and how perhaps some difficult plants might benefit from. He's got some techniques that he uses to sort of get those relationships happening, and that really helps early offspring. So we're often, you know, through Christo, but also through GOC, talking to them about what's important and things like that. We understand that, you know, things like fungi are important. So, but it's not big on our radar, except to say that we will look at it for those functional, you know, aspects. Look, when it comes to Indigenous knowledge, we work with the Juruburu and listen to them as much as we can. We don't want to humbug them too much with things, so we've got our engagement processes that we do every year, and that's important to us and them as well. So, yeah, hopefully that answers the question. Isabel?
Hi. Just curious about, you guys have acid mine drainage problems there?
It's not acid mine drainage. The waste rock is neutral, but the sulfate coming out of that neutral mine drainage has had an impact on Kinjimba Billabong. It's accumulated there and it's caused ASS soils.
Okay. And how this new approach that you guys have now could help with the monitoring of other metals that are under control and being monitored?
Well, I mean, obviously it's a microbial mediated process. There's good and standard national tools for monitoring ASS. So it's something on the work plan for the mine. They're mapping ASS across the site. They're checking for places that it might have occurred and things like that. That's in their future program. They have done some of the work. There's certainly, we've looked at Kinjimba extensively, definitely happened from the sulfate going into that water body. But yeah, it's interesting because it's a chemical approach when it comes to ASS. It's not much genomics that I've seen used for that, but it's a possibility. I guess the CRC Time project is about acid mine drainage rocks. So they're looking at acidic rocks. So it's really good to stay in touch with those sorts of projects to find out what they might learn. But they're looking at it from a management perspective, where if we can control the microbial activity of those waste rocks, can we manage AMD better? So yeah, check in on that project. It's on the CRC Time. Sarah Hammer down Adelaide Union's Flinders Union, thank you. She's leading that. Thank you. So yeah, I guess there's potential, yeah, and they're looking at.
Any final questions? No? All right. Well, thanks everyone for coming. That was a great talk, Andrew. We are going to meeting. There's every Friday they have the morning tea in yellow too. So feel free to wander over there and have morning tea, and you can ask Andrew any burning questions. But thanks everyone for coming, and thanks for coming and giving a great talk.